Okay, I invite you, third years biology students at local university, for our last lecture in animal physiology. And today we will look at the physiology of vision, hearing, and the touch response. Okay, so we look at the eye, the ear, and the various sensory mechanisms that are found within the skin of an animal. So there are various senses, I think 5, but of these, the most important through which an organism receives information about its environment is mainly through vision, hearing, and touch. So you either see something, you either hear something, or you either feel that something has touched you. But more than 75% of the information that most living organisms, at least animals, receive about their immediate environment consists of the visual information, so what they perceive using their eyes. Because you can see something even when that thing is silent, you can see something even even when that thing has not touched you. Okay? For you to use your hearing ability, that thing must produce some sound. For you to use your sense of touch, feeling, then that thing must get in contact with you. But you can see something that has not made any noise. You can also see something that has not even touched you. So in the eyes, we have a section that we call the retina. And within the retina, we have what we call the photoreceptor cells. So a photoreceptor cell is a specialized type of nerve cell that is found within the retina, and that receptor, that nerve cell, it has the ability to transmit some light perception. Okay, so it perceives light, then transmits that signal. That's what we call the phototransduction. So that cell will perceive something, form of light, then transmit that light perception down until it gets to the brain, where now the interpretation is made. Okay. So when you are seeing something, light emanates from that object and it's picked by the photoreceptor cells that are present within your eye, the retina. Then those cells are the ones that will now transmit that information down the optic nerve into the area of the brain that will now be responsible in interpreting what that light means. Okay. So there are some organisms that have their vision being impaired. So hazioni vizuri. So you can imagine we are talking about someone who is, for example, blind. Okay? So it means such a person then will live, even when there is light around, that person or that organism tends to live in a world that we can describe as dark. So we have some organisms which are found living in caves which are extremely dark. So by virtue of such ecosystems, then most of these animals will end up losing their eyesight because they dwell in a place where there is no light. Then their photoreceptors now don't transmit any information. So with the time, such animals end up losing their light. They become blind, okay? Just because they live in an area where their eyes cannot perform its function. But for such animals, the ones that are living in permanent dark caves, because they will have lost their eyesight, they will have now a very developed tactile sense of touch. It will be highly developed because they can't see, but they now perceive through touch. And also they will have a very developed hearing mechanism, because it's through touch and hearing that they can now communicate in the way in which they'll be carrying out their normal day-to-day lives. So for humans, the normal range of hearing, the lowest frequency of sound that a human being can hear is around 20 hertz. Then the highest is around 20,000 hertz. So anything that is lower than 20, that is lower than the hearing range, because it has to be between 20 and 20,000. You can hear between that range. So anything that is lower than 20, then we call that type of sound as infrasonic. Infrasonic. So it is lower than the normal hearing range for humans, that is infrasonic. But if it is higher than 20,000, it is more than the normal hearing range, then we call such a sound as ultrasonic. So if the sound is louder than 20,000, then it is ultrasonic. If it is lower than 20, it is infrasonic. So the infrasonic sounds at times are used by animals like elephants for communication. So it means if you are together with an elephant, there are some sounds the elephant will hear, yet you as a human being, you will not be able to hear. Because the elephant has a slightly lower limit for its hearing, normal hearing range. Okay? Then the sounds that are higher than 20,000, the ones we are calling ultrasonic, those ones are used by animals such as bats. Remember, bats live in dark areas and they are active mostly at night. So they need very improved hearing ability. So they can hear sounds that are higher than the normal hearing range for human beings. Okay. So the ear, and in this case is the animal ear, is not only responsible for receiving sound, but it is also responsible for balance. Okay? It's also responsible for equilibrium, for balance. And we will see the various parts in the ear and what they do. So let's begin with the physiology of vision. So we begin by looking at the eye. So anytime we are talking about the terrestrial vertebrate eye, which means this is eye that is found on a vertebrate that is living within the terrestrial ecosystem. Not in water, but on land, basically. So when you look at the eye of any animal that is found living on land, when light hits that eye, that light begins to be focused in two stages. As we'll see the diagram of the human or the animal eye, when the light hits the human eye, it meets the first bit of aqueous solution. We'll have a look at it and talk about it more when we look at the diagram. So once it gets into the aqueous solution, it is bent. That light begins to be bent or refracted. Okay? So the direction of that light is bent, and bending that light moves it through the surface of the eye that we call the cornea. Okay? So as the light which has been bent after interacting with the first aqueous environment in the eye is bent towards the cornea, just beyond the cornea, again it passes through another aqueous solution where it is refracted again, and it passes through the second structure, now that we are calling the lens. Then it is now the lens that will focus that light to ensure that that light now falls on the retina. Okay? So the light is coming from outside, hits the eye, gets into an aqueous environment, focused through the cornea, refracted again, passes through the lens, fine-tuned, focused and refracted further so that it can now fall on the retina. Then once on the retina, that is now where the image will be formed. Then the image that is formed of the retina, it is in an upside down position. So that image is picked up by the optic nerve, alafu inapelekwa kwa ubongo. So it is your brain that changes the position of that image, which was initially upside down in the retina. But your brain changes it now to be in the correct position. Otherwise on your retina, huwa una image inatengenezwa kama miguu iko juu na kichwa iko chini. Lakini as the photoreceptors that are on the retinas, they pick that information, transmit it to the optic nerve all the way into your brain, then the brain now changes the orientation. So miguu inarudishwa chini, kichwa inakaa wapi? Juu. Okay? So the eye has 3 layers. Okay? The eye has 3 layers. The first layer is what we call the sclerotic coat. So this is a tough layer. Okay? It's a tough layer. And at times people refer to it as the white section of the eye, apart from just the front, where it creates a transparent point that we now refer to as the cornea. So the sclerotic coat is tough, offering protection. Then it is sort of whitish in color, except the front area of this sclerotic coat, which now creates a section that is transparent, and it is that section that we call the cornea. So the cornea, kazi yake is to admit light into the interior of the eye. So light ikitoka nje inaingia kwa macho yako through the cornea. As it gets through the cornea, inaingia sasa ndani ya macho. The first section is aqueous. Refraction or bending of those light rays begin. Okay? Light is admitted into the interior of an animal eye through the cornea. Immediately past the cornea, that light hits an aqueous environment. Okay? That aqueous environment, kazi yake is refraction. In terms of seeing, is refraction. Is bending that light wave. Okay? So it bends those rays. Then as those rays are bent, they now go through the lens. Okay? Then further onto the retina, as is repeated in the second aqueous layer. So the surface of the cornea is kept moist because it cannot be dry, it needs to be kept moist. Na kazi ya kufanya hiyo surface ya cornea ikuwe moist inafanyiwa na tear glands, the tear glands. Okay? So the tears, we always have them because those are the tear glands. They secrete this liquid, this fluid. Then that fluid helps in keeping the cornea, the surface of that cornea, moist and dust free. Okay? So the second layer is the choroid coat. So this is the middle layer, and it happens to be deeply pigmented with melanin. Melanin is dark. Okay? So then, as you would expect, this choroid coat would be dark. So because of this dark color, courtesy of melanin, then the choroid coat will now help to reduce the amount of reflection. Should there be any stray light within the eye, the choroid coat is dark because it has melanin, and that dark pigmentation helps in the reduction of reflection of any light that is within the eye at that particular point. So in the front, the choroid coat now forms what we call the iris. Okay? The iris as well is pigmented. People say this one has dark eyes, this one has blue eyes. So it is the color of the iris that gives you the color of your eye. If you have a black iris, you'll have black eyes. If you have a blue iris, then you'll have a blue eyes. Okay. So the size of the opening of the iris, which is now the pupil, changes, varies, and you don't decide that you want to increase the size of the pupil of your eye, but it is controlled by the autonomic nervous system. That is something that happens without our control. Okay? We don't have control about the autonomic nervous system. So for example, if you are in dim light, ukiwa mahali penye kuna giza giza, the pupil will open wider so that more light can get into the eye. But if you are in an area that is too bright, then your pupil will reduce its diameter. Okay? So by reducing its diameter, it means it is reducing the amount of light that is entering the eye. Also by reducing the diameter, it is helping in focusing, improving the image that is formed. Wale watu wametumia torch. At times you want to focus so that imulishe vizuri. Okay? So ukitaka imulishe vizuri, unapunguza. Ukitaka imulishe pali pafupi, unaongeza. The way we adjust the torches. So the pupil also behaves in such a way. The third layer is now the retina. The retina is the inner layer of the eye. This inner layer of the eye that we are calling the retina contains light receptors. Those light receptors, they can either be rods or cones. Okay? So the rods are cylindrical in shape, but the cones are pyramidal. So the rods appear like cylinders, then the cones appear like pyramids. So each eye will have 100 millions or thereabouts of rods and around 7 million cones, meaning we have more rods than cones. So these rods, they contain a pigment that is called rhodopsin, while the cones contain another pigment which is referred to as iodopsin. So the cones are responsible for bright light vision, ama mahali ya rangi, different the color vision, perception of color, okay, ama kuona vizuri, the quality. So when you are in a brightly lit area, at times unaona mtu akitaka kuona kitu asema na hii ni rangi gani, anatoka kwenda kuangalia huko nje penye kuna more light. That is the function of the cones. Because cones, you use them more when you have bright light or when you now want to recognize color. Okay. But in the opposite, when you are in a dark area, when the amount of light present in a particular area is very low, then rods are called into action. So rods zinatumika when you are trying to see, but the amount of light is very low in that particular place. On page 3, we now have a diagram of the eye. As you can see, we have the cornea here. Then immediately after the cornea, you have the aqueous body. So light ikikuja inapita hapa kwanza kwa cornea, inaingia into the internal part of the eye. Ikifika hapo inakutana na the first aqueous body. Here it is refracted, inakuwa bent. Those the light rays zinakuwa bent or they are refracted towards the pupil. Then ikipita sasa kwa pupil inaenda kwa lens. You know, the pupil is just a section, a tiny section at the center of the lens. Okay. Then now beyond the lens, inaingia tena kwa another aqueous body, the vitreous body, an aqueous environment hapa. So it was bent here, ilipita kwa cornea, ikaingia hapo kwa the aqueous body, it was refracted, ikapita kwa pupil, then ikapita kwa lens, ikaingia kwa the vitreous body. This one also helps in refracting it further so that iende ikue formed kwa retina. The retina is here. Okay? So this section of the retina iko na an extensive abundance, an extensive network of photoreceptors. You have cones, you have rods hapa. And all those are served by nerves, the photoreceptors. The nerve cells. Hizo ndio zina pick the image yenye iko formed kwa retina. They are picked and transmitted through the optic nerve. Inapelekwa kwa to your brain. It gets to the brain for interpretation. Okay? So the image that forms on your retina, it is your brain that will help you to interpret so that you can recognize. But the light that is coming from that particular organism, that particular object that you are seeing, inapita kwa cornea, inapita kwa the kwa the aqueous body, inapita kwa the pupil, the lens inasaidia kufocus, inaingia tena kwa vitreous body. More refraction, inaenda inatengeneza kwa retina. Kwa retina hapo kuna cones, kuna rods. Then hizo zote ziko served richly by the nerves. Nerves will pick that signal, then send it down through the optic nerve to the brain for interpretation. Okay? So through such, then you are able to say, nimeona kitu. Okay? You are able to say, nimeona kitu. Because light, ilitoka kwa hiyo object, ikapita kwa cornea, ikapita kwa the aqueous body, ikapita kwa the pupil, the lens ika-adjust, ikapita kwa the vitreous body, ikaingia, ikakuwa refracted, ikatengenezwa kwa retina. Ikakuwa picked up by the nerves, ikasanywa, ikapita through the optic nerve, ikapelekwa kwa brain for interpretation. So if you have a problem, for example with the optic nerve, then whatever forms on your retina cannot be taken to the brain. If you have a problem with your cornea, then light cannot pass. Okay? So those are some of the problems that someone will eventually have. Ukidungwa kwa mfano macho, so that this fluid itoke, ama hii fluid itoke, then the ability of your eye to refract the light yenye inapita inakuwa ni ndogo. Okay? So that is about seeing. But what about hearing now? Like what you are doing now? Because if you are listening to this video, of course you can see, but most importantly is you'll be able to hear. So when someone says that I'm hearing, it means his or her ears are able to transform the sound vibrations in the external environment. So those sound vibrations are transformed into nerve impulses which are conveyed to the brain. Then the brain now interprets them. So there will be a sound vibration in the environment. But that sound vibration has to be picked, then transformed into impulses by your nerves. Then those impulses within your nerves ndio lazima zikuwe conveyed, zipelekwe mpaka kwa brain. Then it is your brain that will now interpret them as sound. Okay? So it was just a vibration in the environment, but it was picked up, it was transformed. Then it was relayed or conveyed to the brain, and now the brain has interpreted them. Okay? So interpretation ni kazi ya brain. So most of these sounds, they are products of vibrations za objects. So when an object vibrates, then a sound is produced. Okay? So when that object vibrates, for example, if you are plucking the string of a guitar, it will produce pressure pulses within the air molecules. Okay? So that pressure pulse, so that the air molecules begin to vibrate, those ones are what you call the sound waves. Okay? So sound waves are just vibrations of the air molecules. Okay? Then now, the ear, be it a human ear or any other animal ear, has that ability to distinguish different aspects of a sound. For example, the pitch or the loudness. Is it too loud? Okay? So it does that distinguishing by detecting and analyzing the different physical characteristics of the waves. So your ear receives that vibration, that sound, then it is able to distinguish. You say, oh, that is too loud. Okay? Because your ear has distinguished that particular characteristic. So when you talk of pitch, pitch refers to the perception of the frequency of the sound waves. So when we are talking of frequency, it just means how many wavelengths can pass a particular fixed point in a unit of time. For example, how many can pass within a second? How many can pass within a microsecond? If the pitch is high, then it means many are passing. If the pitch is low, then fewer are passing. Okay? When you talk of now frequency, frequency is measured in cycles per second. Okay? So how many cycles can pass a particular fixed point in a second? Such cycles per second have an SI unit that is called the hertz. Okay? So the human ear, for example, is most sensitive and will mostly detect frequencies of 1,000 to 4,000 cycles per second. Anything lower or anything higher might not be perceived easily by the human ear. Okay. Then now loudness. Loudness refers to the pressure that is exerted by the sound waves on the tympanic membrane. The tympanic membrane is what we call the eardrum. So if there is a lot of pressure from a particular sound wave that is hitting your eardrum, then you say this sound is too loud. Okay? So loudness is just how much pressure is being exerted by that particular sound, by that particular wave on your eardrum. If there is very high pressure, then that sound is loud. If there is no high pressure, then that sound is not loud. Okay? So the intensity of that particular sound is measured in what they refer to as decibels. Okay? What they refer to as decibels. So that sound can be transmitted from the environment into the brain or the central nervous system, that energy, that sound energy, should undergo 3 transformations. The first, the sound is happening in the air, so it's eventually a vibration of the air. Okay? So first, the vibrations of the air are converted to vibrations of the tympanic membrane. The tympanic membrane is your eardrum. Something should happen in the air, then it has to be picked, iingie kwa masikio mpaka iende igonge your eardrum, so that your eardrum can now vibrate. Once the eardrum vibrates, then it leads to the vibration of the ossicles within the middle ear. Once these ossicles also vibrate, they will lead to vibrations in the fluid that is found within the cochlea. Then once the fluid in the cochlea vibrates, then there are hair cells within what we call the organ of Corti. We'll see that organ of Corti when we look at the ear, the structure of the ear next. So once the fluid vibrations have been set, they now travel and lead to the stimulation of the hair cells within the organ of Corti. Then the hair cells that are now have been stimulated will now convert the sound vibrations into nerve impulses in the fibers of the cochlear nerve. Then the cochlear nerve sasa hiyo ndio inapeleka kwa brain. Okay? So the cochlear nerve inapick hizo nerve impulses from the hair cells within the organ of Corti. Then that cochlear nerve sasa inapeleka kwa brain, then it is in the brain where the ultimate interpretation happens. So it is only when the nerve impulses have reached into the brain, that is when whoever it is that was listening can now become aware of that particular sound. Because once they reach the brain through the cochlear nerve, then the brain now does what? Interprets. Okay? The part of the brain, the area within the brain is the cerebral cortex. Hiyo ndio inafanya the interpretation. Okay? So it is until those nerve impulses are transmitted all the way to the cerebral cortex, hapo ndio sasa kuna hiyo interpretation. Useme oh, ni umbwa ama ni gari. Okay? So this is the structure of the human ear. As you can see, it has the outer ear. The outer ear is what you can feel ukishika na nje. Then it has a middle ear, then it has the inner ear. Okay? So the outer ear, you have this sort of the funnel-shaped bit. Kazi ya hiyo is to trap the waves. Then it now conveys those waves towards the inner part through the ear canal. Those waves, the vibrations in the environment, the sound vibrations, the air vibrations, which now create a sound, zinachukuliwa, zinapita kwa ear canal, zinaenda zinagonga, they begin now vibrating your tympanic membrane, what we are calling the eardrum. Once your tympanic membrane vibrates, remember, there are these three bones, the malleus, the incus, and the stapes. The malleus imejishika kwa eardrum. Then malleus imeshikana na incus nayo imeshikana na stapes. So once the eardrum vibrates, sababu sound imeingia hapa, itafanya eardrum vibrate, then that eardrum, its vibration will lead to the movement of the malleus, the incus, and the stapes. Okay? Then remember now, as these ones vibrate, they transmit that vibration into the inside of this cochlea. Okay? Into the inside of this cochlea. So once the fluid that is found inside here also vibrates, okay? So that's fluid vibrating because the malleus picked the vibration of the eardrum. Ikaisukuma kwa incus. Incus ikasukuma kwa stapes. Stapes, as it vibrates hivi, inasukuma that vibration, that motion, iende kwa fluid ambayo inapatikana wapi? Hapa ndani. Then as that fluid moves, hiyo fluid sasa ndio itakuja kupata the hairs in the cochlear nerve. Then as those hairs move, ndio zinakuwa zinatransmitiwa into nerve impulses, alafu zipelekwe wapi? Kwa cerebral cortex. Okay? Kwa the cerebral cortex. So let's proceed. As we've seen, the ear is an organ for hearing and balance. What I've explained here was the hearing aspect. But there are these semicircular canals, these bones. These ones are responsible for balance. These ones are responsible for balance. Okay? These ones are responsible for balance. Venye ziko hivi, zote they are in the three hemispheres. Okay? So they are responsible for balancing all those three angles. So these are responsible for balance, but hii ingine yote is responsible for sound perception, that is here. So the external or outer ear consists of the pinna or the auricle. The pinna ni kitu yenye mtu anaweza shika kichuna, unasema unachuna masikio, whatever you can hold from outside. Then the tube or the hole or the canal Okay, that is now what is connecting the outer ear to the inside of the middle ear. The tympanic membrane is the eardrum. That one now divides the external ear from the middle ear. Then the middle ear consists of the ossicles. The ossicles are the three bones, the malleus, the incus, and the stapes. There is also the eustachian tube. This eustachian tube is a canal that links the middle ear to the back of the nose. Na hiyo kazi yake ni kusaidia ku-equalize pressure. Pressure ndani ya masikio ikikuwa too high, it can rupture your ear drum. Okay? So it needs to be equalized. Na hiyo ndio kazi ya the eustachian tube. Then now the inner ear consists of the cochlea. The cochlea is responsible. It has the nerves that help in hearing. It also consists of the vestibules. The vestibules, those are the have receptors for balance. These are the vestibules. These canals, the semicircular canal, those are the vestibules. Okay. Then they have kazi ya balance. Okay. So the question we ask now is, how do we hear? Ni kitu gani inatendeka sahi ndio usikie nikiongea? Okay? So when I talk, your ear, the funnel, the pinna, it traps the sound waves. Then the sound waves that have been trapped by the pinna, the flap ya nje hii, zinaingia kwa canal. Hiyo shimo yenye inatoka nje ikiingia kwa masikio. So ikiingia hivyo, hiyo sauti, hiyo sound waves, they go and now cause your eardrum, your tympanic membrane to vibrate. Once the tympanic membrane has vibrated, it causes the three small bones to move. Remember the stapes, the malleus, the stapes, and the incus, easy tattoo. Okay. Once your tympanic membrane or your eardrum vibrates, it causes these three bones to move. Okay. As those three bones move, the last bone, which is now the stapes, as it moves, it causes wave-like movements to be generated in the fluid yenye inapatikana ndani ya cochlea. Okay? So ndani ya cochlea kuna fluid, na pia ndani ya cochlea there are tiny hairs. So as that fluid moves ndani ya cochlea, sababu stapes imemove, hiyo fluid ina move hivi, hiyo fluid ndio itafanya those tiny hairs to be stimulated. Okay? So those hairs will move, will respond to the different sound frequencies. If it is very high, they will respond differently. If the frequency is low, they will respond differently. So once those hairs are stimulated, the stimulation of those hairs ndio ina generate nerve impulses. Once the hairs that are now within the cochlea have been stimulated, that stimulation makes those hairs now to generate nerve impulses. Then those nerve impulses are transferred into the auditory nerve. They are picked up by the auditory nerve. Then the auditory nerve now transfers those nerve impulses into the brain hearing center, what we call the auditory cortex. So kazi ya the brain hearing center ndio sasa it converts the nerve impulses zenye zimepelekwa huko na auditory nerve. So hiyo ndio ina convert so that uweze kutambua sauti ni nini, ndio ujue hiyo sauti nimesikia ni gani. ni lugha gani huyu mtu anaongea. Ni gari inanguruma, ni umbwa inabweka, ama ni mvua inanyesha. Okay? So sound kutoka nje inaingia kwa masikio yako, inakuwa trapped na pina, inapita kwa the ear canal. Ikiingia ndani, it causes the eardrum to vibrate. As the eardrum vibrates, it makes the three bones, the ossicles, all of them to vibrate. As the last bone, the stapes, as it vibrates, the vibration of the stapes ndio itafanya fluid ambayo inapatikana ndani ya cochlea, hiyo fluid vibrate, isonge, so that the fluid moves. The waves, the movement of that fluid ndio inafanya the tiny hairs that are found inside the cochlea to be stimulated. Once those tiny hairs are stimulated, they begin to generate nerve impulses. So those nerve impulses that are generated by the stimulated hair cells within the cochlea, hizo ndio zinachukuliwa na auditory nerve na zinapelekwa kwa hearing center, kwa akili, kwa brain. Ikishafika hiyo nerve impulse kwa brain in the auditory cortex, hapo ndio sasa interpretation inafanywa. Ndio ujue nimesikia hii, huyu amesema hii, huyu amesema hii. Okay? So that is the process of hearing. Okay? I hope it is clear how hearing happens. From the point where there is a vibration in the environment to the point where that impulse now reaches the hearing center of your brain, so that now you can identify, you can recognize that particular sound. Okay? So that is about hearing. We discussed about seeing. So what about touch, the tactile response? Okay. So there are mechanoreceptors in most animals, and these mechanoreceptors, those are the ones that now help to perceive the tactile response, ile kushikwa, the touch. Okay. So those receptors, the mechanoreceptors, which are found in the skin, they can be described as either being encapsulated or unencapsulated. If the nerve ending is free, then it is unencapsulated. Okay? And the unencapsulated mechanoreceptors, hizo ndio mingi. Hizo ndio zinapatikana in many parts of the skin. Okay? So some of these unencapsulated nerve endings, they happen to be sensitive to either some painful stimuli, kitu kikudunga ama ikikuuma, so the change in temperature, whether it's too hot or cold temperatures, and to touch, even if it is very light. That's why mtu akikuguza kwa kidogo unaona, dudu ikikutembelea you feel. But that touch is extremely light. Okay? That touch is extremely light. So those mechanoreceptors, they sense the stimuli because their plasma membranes are deformed in the process. So when there is a touch, that touch leads to the physical deformation of the plasma membranes of those mechanoreceptors. So those mechanoreceptors, their plasma membranes, they contain ionic channels which are mechanically gated. Okay? So if there is a touch, then that, because the touch is mechanical, so that touch can either, that mechanical touch can either lead to opening that ion channel or closing that ion channel. That's the meaning of the mechanically gated ion channel. So, talking about the human skin, there are 4 primary tactile mechanoreceptors. We have the Merkel's discs, the Meissner's corpuscles, the Ruffini endings, and the Pacinian corpuscle. So there are 4. 2 of each are located towards the surface of the skin, and the other 2 are located deeper into the skin. So the Merkel's disc, for example, and the Meissner's corpuscle, they are located towards the epidermis, towards the surface. But the Ruffini ending and the Pacinian corpuscle, they are located deeper. Okay, they are located deeper. The Merkel's discs, these are found in the upper layers of the skin, and they happen to be present whether the skin has hair, kama kwa kichwa, ama the skin is glabrous, meaning it is hairless, kama ndani ya mkono, on your lips, in the sole of your feet. So hapo bado utapata kuna Merkel's discs. Okay? So you find these Merkel's discs to be so densely distributed at the tips, at your fingertips, and at your lips. So it means that the tender touch that makes you to be aroused, either on your lip, either on your fingertips, that is courtesy of the Merkel's disc. So this Merkel's disc, they will respond to light touch because they are not capsulated. Okay? So they will respond to very light touch. Okay? So the fields that are used by the Merkel's disc to perceive the stimuli, they have very well defined borders. So that's why ukishika kwa kidole unaweza jua nimeshika kando ya kidole, nimeshika katikati, because those Merkel's discs, ukishika hivi, those are the ones that are perceiving that touch. Then, because their borders are very well defined, then you can sense nimeshika kando, nimeshika wapi, na hiyo kidole tu. Okay? So, this Merkel's disc, they will respond to very fine touch. Very fine, the light one, very light touches. Okay? They are found mainly at the fingertips and on the lips. The same Merkel's discs and the Meissner's corpuscles, they are not found in plenty in your palms, ama kwa mkono, okay? But they are found mainly at the tips. As you go deeper into the skin, ukienda chini sasa unakutana the Ruffini endings. These Ruffini endings zinapatikana pia kwa ile ngozi ambayo iko na nywele na ile ambayo haina. So you can find it on your head, you can find it in the soles of your feet. Okay? So these ones are encapsulated, which means they don't respond to ile fine touch. So kwa mguu in the sole, unaweza kanyaga na pressure sababu unatembelea hapo. So the perception you receive, kama kuna kitu inakudunga, utaipata sababu ya the Ruffini endings. Okay? These are encapsulated. So zinahitaji pressure kiasi, not the fine touch yenye inapatikana kwa the finger tips. Okay? So these Ruffini endings zenye ziko nao deeper, hiyo zina detect warmth. Okay, zina detect warmth. So because the Ruffini endings, which are responsible for detecting warmth, zinapatikana deeper into the skin, inamaanisha what can help to detect cold inapatikana nje, then what can can help to detect warmth nayo inapatikana deeper. So then, as a human being, you end up having the ability to detect cold earlier before you can detect warm. So if there is a change in temperature, ikianza kuenda chini, you will sense faster kuliko kama inaenda juu, because what detects cold inapatikana on the outside, but what detects warmth, the Ruffini endings are found deeper into the skin layer. The Pacinian corpuscles, which are also located deep in the dermis, happen to be found as well in a skin which has hair and in a skin which is glabrous, meaning it is not hair. Okay? So equally, they sense deep transient pressure. So something that is not prolonged. You are pricked by something. Okay? Sio kitu ambayo inafinya for long, but it has to be deep and not prolonged. Okay? So the Pacinian corpuscles will sense that. They will also detect pressure and vibration yenye inaletwa because of the compression. Okay? So they detect vibrations, they detect pressure. What is important? What is important is that there are fewer Pacinian corpuscles and Ruffini endings in the skin, but there are more, the Merkel's discs and the Meissner's corpuscles. So how you perceive anything that touches you is a factor of those four mechanoreceptors. Is it a Merkel disc? Is it a Meissner's corpuscle? Is it a Pacinian corpuscle? Or is it a Ruffini ending? Look at this diagram on page 8. It will show you where they are located. Okay, the Merkel's disc, the Meissner's corpuscles inapatikana towards the surface of the skin. The Ruffini ending and the Pacinian corpuscle nayo inapatikana ndani, further deep within the dermis. The Merkel's, the corpuscle, the Merkel's disc, the Meissner's corpuscle, they respond to fine touch. They can sense cold. The Ruffini ending can sense warmth. Okay? The Pacinian corpuscle, deep. The Ruffini ending, the Pacinian corpuscle hazipatikani mingi sana kwa ngozi in comparison to the number of the Merkel's disc and the Meissner's corpuscle. Okay? So that is about how we sense touch. How we sense touch. The touch, the tactile response inakuwa picked either by the Merkel's disc, the Meissner's corpuscle, the Ruffini ending, or the Pacinian corpuscle. How or what picks it is a characteristic of which stimuli. Okay? Which stimuli. So, read through this, listen to these explanations, and should you have a question, you can always reach out. Okay? Otherwise, for now, that marks the end of our lecture today. Thank you very much, and let's keep in touch. Thank you.